So I don't want to cover too many things that were covered by some great researchers doing some really cool things in a lot of different areas today. So I'll kind of focus a lot on some engineering things, talk about myself a little bit. But yeah, so who am I? I'm a scientific consultant working at Brain Vision. My background is in physics and engineering, and I more recently have a PhD in neuroscience. My research focus in neuroscience has been primarily on BCI, using EEG signals, mostly for replacing communication for advanced ALS patients, and a lot of different things kind of surrounding that. So recently at Brain Vision, among many other things, one of my favorite things going on is in non-invasive neuromodulation and EEG, and how the combination of those two things is informing on brain circuitry and providing some new areas of treatment for clinical populations. So I'm part of a team of ex-researchers at Brain Vision that are helping out a lot of different labs outfit their lab with new technology, new capabilities to do new research. That's pretty much what we do. And we work with a lot of different manufacturers of equipment. Here's a handful of them. But we're always looking at what's new, trying to find out what researchers are looking to do, what sorts of things have they not really been able to explore yet because of some sort of technological capability that's not really there yet, and see if we can't bring new ideas out into the research community and take as much as we can from the research community and lessons that are learned and give that information back to these and other manufacturers so that easy to use technology is there for a lot of different labs to do some cool science. So I'll talk a lot about EEG today. A lot of the things that we do are all really non-invasive. So we don't get too involved with the technology around doing single unit recordings, local field potentials, and things that require, surgery, I guess, basically. But when you think about getting from, the actual tissue that's creating this activity, actual neurons firing, the next closest thing when you talk about getting outside of the actual body of the nervous system is MEG and EEG. Dr. Xiang talked a lot about the two of these modalities, the differences, and some of the benefits and combinations that are really good. You get a little bit further away, you look at correlates of neural activity, which, most of these are basically just about blood flow changes in the brain based on activity. And you can look further away at peripheral signals, and then you get all the way to the point where you're just exploring human behavior. And of course, there's pros and cons to all of these things, and exploring all of that stuff. And so talk a little bit more just about the ideas of where EEG is coming from. I think a little bit of this was discussed today, so I won't go too far into it, but effectively, right, Dr. Xiang also pointed out, we're talking about changing a voltage on the scalp really can only be measured by these techniques, EEG and MEG, when we're looking at tens of thousands, if not millions of synapses firing simultaneously. Those magnetic fields and electrical fields and currents that are moving through the neural tissue and finding their way through the skull and outside of all that tissue out into the scalp where we can measure it non-invasively. It's a very interesting concept about what all has to happen for us to be able to measure any of this brain activity. I think it's important for researchers to understand this. This is a little bit of an excerpt from Steve Luck's book from 2005, which is also a great resource. And then again, looking at EEG and MEG, I think a really interesting thing to think about with those two modalities is what can be measured with EEG or MEG is really based on the orientation of cells in a gyrus or a sulcus. And so there's a lot that these two modalities can complement each other, depending on how these circuits are oriented on the cortex. So one big consideration for applying this technique is how do we align and put all these sensors near the brain. And so in most cases, you're applying EEG sensors in a standard fashion, which we usually call the 10-20 system. And this 10 and 20 are referring to spacing, basically a percentage of the head circumference that we space each of the electrodes out. You can do something like this with EEG and MEG, and we end up doing it with FNIRS. The other kind of major thing that's used pretty often is equidistant montages, so laying everything out so that they're all equally spaced, not only in a direct chord across the scalp, but basically just trying to space everything out equally. And that's a technique used, and that can be kind of helpful and beneficial in source analysis later on. So another thing I'd like to point out about looking at all these different modalities is something that was pointed out too by Dr. Shang, and that was about the spatial and temporal resolution of these measurements. And if you think about putting together things like EEG and MEG that have great time resolution, and other techniques like fMRI that give you great spatial resolution and additional information that might be captured in a different way, you see you can really capture, when combining techniques together, so much information about the time and space that different neural activity is occurring. So we do a lot, and we're really happy about the solutions that we provide when putting together things like EEG and fMRI. And we have lots of labs that we work with that do EEG and MEG together. If you're not wanting to do something inside the MRI, then NIRS is another example of being able to do something reflective of the hemodynamic response, and you can do that in different scenarios and different environments. So I'm going to talk a little bit just about EEG technology, kind of where it's at now, where it's come from to some degree. The first place that I think that is really important and that's helped out researchers be able to do a lot of different types of experiments is in active EEG electrodes. So, traditionally EEG electrodes were passive in the idea that this is just a conductive piece of metal that's held onto the scalp as close as possible. The scalp is electrically connected to the EEG electrode through some gel, and whatever tiny microvolt changes occur on the scalp are measured far down a length of wire to a set of electronics that are amplifying and digitizing those voltages rapidly over time. So with active electrodes, we're taking technology and really great, miniaturization of electronics and putting small amplifier circuits in each of the sensors themselves. So it gets us measuring the EEG actually right at the scalp a little bit more so than with passive electrodes. And this has done a huge service in being able to record in noisy environments, getting EEG recordings outside of a shielded booth in a shielded room, and in more real world, lifelike scenarios for research. Saltwater sponge electrodes have been around for quite a few years, and a lot of companies, including Brain Products, have adopted making these and integrating them with their systems. They've provided, a very quick and easy setup for EEG, a way to apply a lot of electrodes in a very short time. It's been very popular with researchers working with kids and populations that are a little bit more sensitive about having something on their head, like ASD patients and things like that. And then there's just been a lot of customization that's possible with whatever your application is for recording neural activity in both the cap and the electrodes and just how everything is arranged. This is just an example of a very small amplifier that's in the actual cap fabric, a handful of electrodes, which makes this very easy to record EEG in highly ambulatory and mobile situation. The other thing that's been around and has been becoming more and more pervasive in EEG research is dry electrodes. And there's a good number of companies out there creating some pretty neat electrodes that don't require gel. You don't have to have gel in people's hair. It's somewhat similar to the sponge nets in that you can quickly and easily put a headset on like this. This is, again, a totally wireless mobile technology. These wires are just going to these ear clips for ground and reference connections. And then a host of different sensors that provide a different level of comfort and appropriateness for how much hair is there. So just flat things for maybe forehead and parts of the scalp that don't have hair. And then dry sensors that can get through hair and also provide good signal. So the other thing I've already kind of mentioned a little bit is about taking EEG more mobile. The system that I have here and that I have pictured here is a little bit more similar to the typical laboratory setup, a box with electronics in it, connected via wires to the EEG electrodes on the head. This one is battery powered, and this box would sit stationary. The participant is likely to be just sitting stationary, viewing a computer screen, going through some task, or presenting stimuli to the participant for whatever the research paradigm is. And this device, it does a lot of great things. It's got active electrode technology integrated into it. It's got a number of additional channels that you can record other physiological measures at the same time. Allows for very clean recordings because it's battery powered, and a lot of other advancements in the electronics. But now we've miniaturized all those things and are allowing people to do experiments when people are not just sitting stationary at a computer screen. They can go out into the world. Biking has become a pretty big thing. Lots of different exercise and sports science have utilized these things. A lot of labs that are looking at motor recovery after stroke and things like that can look at gait and other types of behavioral analysis along with EEG, and that's been pretty helpful. And this is a 64 channel setup, but we've even seen some nice active electrode technology embedded in a 128 channel system. So the application space is pretty wide. The types of amplifiers and devices out there are specialized for a lot of different areas of research, from lots of different participant populations. And here's an example of TMS and EEG. So I wanted to sort of here's a giant list of all the types of research fields that we work with pretty often and that we see out there. More traditional psychology and psychiatry, clinical applications. We work with business school professors that are looking at neural activity that's based on advertising and neural marketing types of things, neuroergonomics. We talked a little bit about BCI applications today as well. And MOBI is sort of this general idea of doing EEG and doing non-invasive neural activity monitoring while people are doing real world, real life kind of activities. And the thing that I think is also exciting is just how many different modalities you can apply along with EEG that can help inform more on what is this neural signal that I'm looking at doing, and capturing additional data that sort of complements what you can learn from just the EEG signal. So I was just going to talk a little bit about the general ideas, if anyone's new to EEG, about what are some key considerations for applying this whole technology. Having a cap lets you have electrodes placed in these standard positions that I mentioned previously, put them on the participant's head without having to measure necessarily every single sensor. So it really allows you to rapidly put electrodes in place and maintain their position and their electrical contact with the scalp. it's important to get that fit in the position of the cap just right, so that you have all these electrodes in their standard positions, so that you have the least amount of inter-subject variability based on where the cap is at, because you're going to get some regardless, just because of anatomy changes and differences between people. And then it just provides you an easy way to apply and remove those electrodes. And then applying additional measures at the same time and making sure that you've got good impedance is basically the key thing when preparing electrodes. And there's a technique to it. It's sort of an art. There's not much that you can do besides getting a little bit of practice in. I prepared a couple of electrodes. We'll see how good the impedance is on them here in a minute beforehand. But you'll see that it can make a big difference in your signal quality, and it's something that is dependent on the researchers themselves when they're applying things. And the technology can't do much more for you unless you get in there and apply gel in an appropriate way and deal with hair. And a lot of different people and a lot of different subjects will have all sorts of different considerations for doing this as best as possible. So we've just gone through a whole brainstorm tutorial and looked at a lot of different things like artifacts, how to remove them. So these types of things that we've seen already are obviously blinks. We see maybe a couple of channels with some extra noise in them, might indicate that they have high impedance. We might see some higher frequency information in a channel like this, not as many bigger amplitude spikes, but still a little bit more high frequency. And then we see a lot more of the saccades that Martin mentioned earlier. So yeah, now I would like to just see if I can show the recording software. Let's see if I just need to change the window that I'm presenting. So this is just the Brain Vision Recorder. This is the software that we use to interface with the equipment from Brain Products in general. Effectively, this is just a way to set up the details. I guess I need to plug in things. I'll go through quickly just how you set up some parameters for doing the EEG recording. The typical, no, I think I have to redo this since I just plugged in the device. Okay. Well, let's just jump into it. I don't have to show those specifics right away, but yeah, the first thing you'll end up doing in most systems, most EEG systems out there will be to run some sort of impedance check to let you know what sort of impedance you're seeing for each of the electrodes. So there's nobody else around because it's COVID times. I've been in the office by myself all day. So I did the best I could to try to gel some electrodes myself. The easiest ones to get to are the ones up here in front. So it actually looks like I'm at pretty close to just a couple of kiloohms on these electrodes in the front, which is pretty easy to get to. I didn't actually apply any gel into a lot of these electrodes out on the side, and in the back would be pretty tricky, but I could probably figure it out. I can mouse over it and see where each of these are. I don't know how easy it is to see in the camera, but each of the electrodes on the cap has a little LED in those preamplifier circuits. And some of them should be green and some of them should be red, and that sort of corresponds to the colors you see on the screen as well. And so that's kind of just a handy little feature in these active electrodes. While while a researcher is preparing and applying gel into each electrode, it gives them an indication if there's a good electrical connection with that electrode already. Once it turns from red to yellow, then you know that you've got a reasonable impedance, and when you get down to green, you know that you've met your criteria for impedance, and you can move on to the next electrode. That also helps speed up adding in gel and preparing the electrodes. So I'll jump back to looking at the data, and since I haven't prepared all the electrodes, I think I should be able to jump to a screen where I'm just looking at some of the electrodes that I've already applied the gel to. So yeah, just here in real time, you can see when I do a couple of very deliberate blinks that you can see some pretty big deflections, particularly in these two channels that are right up on the front. You'll see like left and right eye movements to some degree in there. And let me turn off any sort of display filters as well. So we can just see the raw data there. And you can kind of tell there's not a huge difference between the raw data and something with some filters applied. The other things that show up really big are muscle artifacts. So maybe if I'm moving around and have a little bit of neck muscle tension, if I had some more of these lower electrodes gelled, you might see a little bit more there. Tightening a jaw. Because this is such a big muscle, the masseter muscle, if you have someone clench their jaw, you should see pretty large EMG artifact show up in the signal. And that's one reason why, even with talking right now, you'd expect to see a lot of additional noise in this data. Part of the reason that it looks fairly clean is because these are active electrodes. Part of the reason that you don't see a whole lot of that is a lot of EMG in there is because I've got a reference electrode and other aspects of the technology, but certainly you can see that the amplitude of blinks and muscle artifacts is a lot bigger than EEG. So there's a lot that technology has helped us do to get rid of some of those things and give us a clearer picture of what the brain's up to. Another big consideration for collecting EEG is putting markers into the data. So markers are basically a way of us marking in the EEG data itself when some sort of event typically happens. So ERP types of scenarios. Let's see if I can add this in. So another one of the devices that I have out on the table here, besides the EEG amplifier itself, is something we call a trigger box. So this is a device that's allowing me to send timing pulses with a lot of temporal precision to the amplifier from potentially my stimulus presentation computer or something like that. So if I turn on a little application, I'm not sure how clearly we can see this, but down at the bottom, I'm sending rapidly at regular intervals an array of timing markers. So later on during analysis, as we saw earlier, you'd end up segmenting the data up based on when these markers showed up in the data to allow you to average segments of the EEG data or do other types of analyses with the knowledge that a certain event has occurred time aligned with one of these markers. So there's a lot of different considerations for how you end up getting timing based on what the paradigm is. But items like this trigger box help us to integrate potentially timing information from a lot of different sources, like eye tracking data, like a TMS device, signals from the fMRI if I'm doing EEG and MRI at the same time, things like that. The only other thing I have, and I don't know what my camera looks like now too much. I should zoom this out, but I've also got one of these small mobile amplifiers and a saltwater net sitting out here just for something to look at. But those are the basics I just wanted to run through. I'm happy to talk a lot